Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Katrine Whiteson. Dr. Whiteson is Assistant Professor at the Molecular Biology and Biochemistry School of Biological Sciences in Irvine, California, and Associate Director, UCI Microbiome Initiative. Dr. Whiteson's team showed how dietary shifts impact the gut microbiome by preferentially selecting for microbes capable of utilizing various dietary nutrients. Today, Dr. Whiteson presents her session, High Fiber Whole Food Dietary Intervention Alters the Human Gut Microbiome, but not Fecal Short-Chain Fatty Acids. Dr. Whiteson, over to you.
Thank you very much for the opportunity to speak today for the Microbiome First Virtual Summit. I'm excited to tell you guys about my research. I'm going to tell you about our experience with doing a fiber microbiome intervention in an undergraduate class, and also some of our experiences studying microbiomes from other parts of the world. So when I talk about microbiomes, I'm talking about the collections of bacteria, viruses, fungi, and other microbes that live in a particular environment. We now know, after the last 10 or 15 years of good access to inexpensive DNA sequencing, that human microbiome composition correlates with a number of health and disease states like obesity, cancer, autoimmune diseases, including asthma, brain chemistry, and even vaccine efficacy. And this brings me to a point that now that we have this capacity, you can even pay to have your own microbiome tested. The trick comes down to the interpretation. So our big challenge in the next 10 years is to figure out how to interpret and make use of a lot of this data. And I have a lot of optimism given the recent advances and some of the findings in, for example, outcomes of dietary interventions and the use of probiotics. Certain probiotics, for example, the bifidos that can break down human breast milk sugars, are examples where we see really promising potential for modulating health through the microbiome. However, I appreciate the take coming from this New York Times piece where they acknowledge the limitations in what we know about how to interpret a microbiome now that we know how to sequence them, because there are some important limitations. Today, I definitely want to acknowledge Andrew Oliver, now Dr. Andrew Oliver, a PhD student from my group who defended last year. And he was heavily involved in two of the projects I'm going to tell you about today. He led the undergraduate class intervention. Here he is with the students, and also an analysis of microbiomes from Ecuador that I'm looking forward to telling you about. So I'm zooming to you from the University of California, Irvine. You can see here we are between Los Angeles and San Diego. We're near both the beach and the mountains. We're actually hiring 2 virologists this year. And I just wanted to share my email and point out that I'd be very happy to field questions and to hear from you after the seminar. And I want to make sure I acknowledge my group. Here we are before the pandemic. And again, Zooming. I'd also like to point out that since I started my lab here at UC Irvine in 2014, I was at first inundated with requests to collaborate from all over our campus. And together with an ecologist, Professor Jen Martini, we started a microbiome initiative on our campus, which has been really fun and given me the opportunity to work with a lot of different interesting projects. We also realized that we were not the only campus looking to start a microbiome center. So we've started to pull the center directors from around the country together, and we now have the Microbiome Center Consortia. There are seminars every Thursday at 11 a.m. during the academic year that I can highly recommend. And here you can see us meeting before the pandemic, and actually we just met in Chicago a few weeks ago too. Traditionally, humans and the environment were studied separately when it comes to microbes, but the truth is the microbes don't distinguish. And many ways that we look at microbial communities in the context of soil or ocean have led to principles that have been very useful for also understanding human microbiomes. So while a lot of clinical microbiologists in the last century have focused on one pathogen at a time, the truth is the microbes live in polymicrobial communities, and the communities themselves impact our health. And so we've really benefited, or I know I've really benefited from learning from the ecologists who have been thinking about microbes as parts of communities for a very long time. We now know that each person has a unique and persistent human microbiome inhabiting each of the different environments in our body. Most of the genetic differences between you and another person listening to this seminar are encoded in our microbial communities. That's even true for identical twins. Some of the most dense microbial communities on the planet are living on the human body. And in this plot here, you can see the bacterial counts per volume are quite high for colon or dental plaque samples, even higher than soil, for example. And of course, the density of microbes differs across environments and is lower in some parts of the human body. And for every one of those bacteria, there's usually about 10 times as many viruses infecting the microbes. And we also just have an incredible diversity of molecules. So if I had a very small volume, even a few microliters of blood from anyone listening to this seminar, given enough different types of mass spectrometry instruments, I could detect about 500,000 unique metabolites, and probably about half of those have microbial origins. And now that we've had some time to think through the impact of our microbial colonization, we now know that disease is sometimes arising from a disturbed microbial community. And it's especially important in our era with industrialization and a lot of changes in our microbial exposure. This plot right here is a big part of what convinced the National Institutes of Health to fund the Human Microbiome Project in 2005. So here you can see in the second half of the 20th century, as infectious diseases were decreasing dramatically and lifespan was increasing. This is in large part due to big efforts, big public health efforts, including sanitation, vaccination, and other efforts. We had this big decrease in infectious disease. But during the same period, we saw a profound increase in autoimmune diseases. That includes asthma, type 1 diabetes, multiple sclerosis, Crohn's disease, and actually I think some kinds of cancer may belong here. And so this has led to, in part, to this huge boom in enthusiasm for studying the microbiome. And a big part of it might be that we have these changes in exposure to microbes. For example, if you read Marty Blazer's book, Missing Microbes, you can learn that an average child in the United States is exposed to 18 courses of antibiotics before their 18th birthday. And so that's one of many of the big changes in exposure to microbes that's happening to people who are growing up in the industrialized world. And this is, of course, having a big impact on how our immune systems are developing. This has impact for cancer as well. And for example, there have been some really interesting findings recently from Jen Wargo's lab at MD Anderson, among others, showing that, for example, if you're getting cancer immunotherapy in response to melanoma, for example, if you have a high fiber diet, you can really have a big impact on the chances of your immunotherapy succeeding. The same is true for fecal transplants. If about a third of immunotherapies are succeeding, it turns out that if you can get a fecal transplant from somebody whose immunotherapy succeeded, that will increase the chances of the recipient's immunotherapy succeeding as well. This is one of the things that really inspired us to understand the impact of dietary fiber on the microbiome, because in the study they show that the fiber on its own is really important for impacting successful cancer immunotherapy outcomes. And in our lab, we actually have done a little bit of work following microbiomes in the context of cancer. This is Julio Avalar Barragan, a student in our lab. He's co-authored a review, and he's recently led a study of polyp microbiomes in the context of colon cancer, and looking at the distinct microbes that are associated with each polyp type. And of course, this colon cancer is becoming more common in the industrialized world, and it's occurring in people earlier when they're younger. And this also is something that is related to industrialization and changes in microbial exposure. Here's another really worthwhile study to look at, which is a study of immigrants from Thailand moving to the United States and how this impacts the relative abundance of Prevotella, which is a bacteria that can break down plant fibers. So with every 5 years that somebody spends in the United States, you see this decrease in the prevotella, which are famous for being able to break down plant fibers. And by the time somebody has lived in the United States for 30 years or more, their prevotella abundances come to resemble that of somebody who's lived in the United States their whole lives. And this brings us to the importance of preserving the microbes that have been with humanity through our history. And here's a really interesting effort, the Global Microbiome Conservancy, which is working to preserve the microbes that come from all different environments in the world as they're changing due to climate change and industrialization and other forces. So when it comes to looking to manipulate the microbiome, what are our options? Well, we've got probiotics and prebiotics. So prebiotics being like fibers that are microbial accessible nutrients that resist early digestion and make it to our colon where most of our microbes are. And then, of course, there's probiotics, which are live microbes. Some of them are more likely to promote health, and really they're the gut anaerobes that can break down fibers. And others, many of the ones that are on the market, are actually more adapted to living in yogurt and are not necessarily the gut anaerobes that break down plant fibers. And then we've also got fecal transplants and phage therapy. And for my talk, I'm looking forward to telling you this story about our experience of working with fiber as an intervention. So what is fiber? Fibers are carbohydrate polymers that have these repeating units of carbs linked together, and they are not able to be broken down by human enzymes. So they persist in our digestive tracts all the way to the colon, and that's where most of the microbes live. That has interesting impacts also for blood glucose response and for dosing insulin. So, for example, here's some white bread and some whole wheat bread. And if you are dosing insulin, you can actually subtract the fiber from the amount of insulin that you need to cover a certain amount of carbohydrates. So in this whole wheat bread, for example, you could remove those 4 grams of fiber because they're not going to quickly impact your blood glucose. But amazingly, if you look at the fiber content of beans and lentils, you'll see that it's about half of the carbohydrates that are present in beans or lentils that are in the form of fiber. And Old Navy beans are an important example here. For example, at the MD Anderson Cancer Center, which I just mentioned, Jen Wargo for her previous cancer study, they actually have a clinical trial where the intervention is Old Navy beans. It's called the BGON study. And the outcome is to understand, it's to prevent the recurrence of colon cancer. And they are promoting eating 1 cup of Old Navy beans a day. And if the daily recommended allowance in the United States is 25 grams of fiber a day for women and 35 for men, but in most cases, people in the United States are getting about half of that. If you were to add a cup of Old Navy beans a day, you would really make it a lot of the way closer to the daily recommended allowance in the United States. So just having a look through the digestive tract here, where you see there's an increasing density of microbes through the digestive tract. And by the time you get to the colon, you have the densest habitat for the microbes, 10 to the 9th to 10 to the 11th, mostly anaerobic bacteria are residing there. And if you eat low fiber starches that are quickly absorbed, like rice, you actually don't have a lot of nutrients persisting all the way to the colon where the microbes are. Whereas higher fiber starches like lentils will persist all the way to the colon and give your microbes something to eat. In fact, when you compare people from the industrialized world to people from other parts of the world where they're eating a lot more fiber, you find that in the industrialized world, our guts are enriched in microbes that break down the mucus lining our gut, like Akkermansia is a famous example. And we see all these mucus degrading bugs in our guts because there's not much fiber, there's not much substrate for the microbes to eat. Whereas in other parts of the world, you find enrichment for the bacteria that are famous for carrying a lot of the enzymes that can break down plant fibers. We were very inspired by this study, a diet swap study, where people from the United States and people from rural South Africa swapped diets for just 2 weeks. In rural South Africa, they're eating a low fat, high fiber diet with lots of the beans and lentils and other fresh fruits and vegetables. Without even trying, they're often eating something like 66 grams of fiber per day. Whereas in the study cohort of African Americans from Pittsburgh, on average, they were eating more like 14 grams of fiber a day, which is somewhat consistent with what's going on in the United States. There's much higher fat content in the US diet compared to the South African diet. So, what happens when you're getting a lot more fiber into your colon is that the bacteria in the colon are fermenting that fiber, and they're producing short chain fatty acids. So these are fermentation products, including butyrate. These are volatile molecules that do not smell very good, but they are the favorite fuel of the cells lining our colon. The epithelial cells, for example, they love to run on butyrate. And if you're not eating a lot of fiber, you're basically depriving those cells of their favorite fuels, which has all kinds of impact for our immune health. So, for example, these molecules are known to decrease inflammation and to hinder tumor growth, whereas animal fats lead to the production of secondary bile acids, which are considered carcinogenic. So in this study, there was just a 2-week swap, and they took colonoscopy. They did colonoscopies before and after to have a look at the colon health. And in these 2 weeks, they found that the people eating the low-fat, high-fiber diet shifted their microbiomes towards the fiber degraders, and there was a decrease in inflammation and what was considered a clinically meaningful decrease in markers of cancer risk. For example, they were looking for these little precancerous growths in the colonoscopy. Whereas in just 2 weeks, swapping the diet in the other direction, so the people in South Africa got to eat barbecue chicken and a lot more meat, which apparently they really enjoyed, but they found in just after those 2 weeks of intervention, they found increases in inflammation and markers of cancer risk. And so here's another view of the dietary fiber being broken down through fermentation by the bacteria, and short chain fatty acids, including butyrate, are a really important product, which are, as I mentioned, the favorite fuels of these cells lining the gut, with really important impact on our immune health through a number of different pathways. We were also really inspired by this citizen science project, where they found that eating 30 different types of plants in a week is associated with higher gut microbial diversity. And in this study, while there were 10,000 people in total, they had careful diet data for about 1,000 of them. And so here's my question. So how many of these people do you think were eating more than 30 types of plants in a week? And the answer can be found here at the bottom of figure E. And you can see that there were about 42 people out of the 1,000 with careful diet data who were eating more than 30 types of plants in a week. Of course, this might also correspond to just generally eating more fiber. But I think it's really interesting that they found that higher plant diversity corresponded to higher gut microbial diversity, implying that the microbes were, the higher diversity of plant fibers was supporting a diversity of bacteria that could break down those fibers. Another study we were really inspired by came from the Sonnenbergs at Stanford, and they compared a fiber diet intervention with a diet intervention that included increases in fermented foods. And they found that the fermented foods more consistently increased gut microbiome diversity across the people in their study cohort. And this intervention lasted a little bit longer, and they saw this more consistent increase in gut microbial diversity from the fermented foods. And they mentioned that it's likely to be due to the molecules contained in the fermented foods promoting a diversity of microbes, and not necessarily that the microbes in the fermented foods are themselves able to colonize and grow in the guts. So it's more about the molecules from the fermented foods promoting the growth of the resident microbes that are already present in the gut. So this led us to our question: how does a wholesome diet high in fiber change the gut microbiome and influence the abundance of short-chain fatty acids in the intestine? And so we had the opportunity to lead this class of students. There were 26 students, and we developed a course-based undergraduate research experience, a CURE course, with this 2-week intervention. And here you can see Andrew, the grad student from my lab, with the students in the class, and the instructor was Julia Massamelli. And we really had a lot of fun with this whole experience. We wrote 2 papers about it, an education-focused paper and also a science-focused paper, which are both published and available now. And it's led to some future really fun projects, including funding from the Bean Institute, where we've been cultivating fecal samples from different people with different fiber sources, for example, from different kinds of beans, to try to learn about which bacteria are best able to break down those fibers. And actually, as part of this, I've also been giving lectures together with UCI chef Jessica Van Roo, and I always learn a lot from her. And actually, we have a video that's available on YouTube, and here's the recipe for lentil soup and no-bake energy bites. And so she would be cooking really real time while I give the seminar, and then I'm talking about the results from the fiber intervention. And I always learn a lot from her, and I encourage you to check this out if you're interested. Okay, so there was this recent spring quarter where we had our 26 students enroll in an experiment to eat 50 grams of fiber per day. And we had to fit everything into the 10-week quarter. So we quickly got everybody keeping track of everything they were eating with an app called MyFitnessPal. We had them collect 3 fecal samples in that pre-intervention phase, and then we went for it with the intervention during weeks 2 and 3 of the quarter, starting with a goal of 40 grams of fiber per day, and then moving to 50 grams per day. And then the study ended. We collected 3 more fecal samples, and then we went on to collect the data so that the students would have a chance to analyze the data before the end of the quarter. In order to get to that 50 grams of fiber a day, we provided the students with 2 meals a day from this meal prep company called Thistle, and each of the meals had pretty high fiber. I looked into the meal prep options at various meal prep companies throughout Orange County, California, and these guys had really high fiber. So that got us pretty close to the goal. And then we also counseled everyone on ideas for things to eat that had high fiber. For example, so here's survey data showing you which foods they turned to get their fiber. And I know I learned a lot about what the best foods were to get this fiber. We found that, for example, berries are a great source. They have low glycemic index and actually pretty high fiber content. I didn't know that avocados were a good source of fiber. I knew they were a good source of healthy fats, so I thought that was really interesting. And then, of course, some people were using fiber bars and cereals, and a lot of people were turning to lentils and beans, which, of course, have a really good density of fiber for each carb. So our first question was, just did we succeed in this? Did the students increase their fiber intake? And absolutely, they really did. There were some students starting out in week 1 with literally 0 grams of fiber on average. And overall, it was less than 20 grams a day for the average of the students as a whole. And by the time we got to weeks 2 and 3, you can see we really did get the fiber grams per day up above 40 into 50 per day. And then we did shotgun sequencing to examine the microbial community composition and function. And everything's been published in this paper in M Systems, so you're welcome to get the full story there. A really important signal which emerges in any microbiome study is that there are really strong individual signals. So here I'm showing you the relative abundance. Each of these columns represents one person, and the different colors come from the most abundant microbial taxa. And you can see each individual is clustered together here, and the samples from each individual have a lot of common features. So that is the strongest signal in the study for sure. But we do see these really interesting changes. So in gray is the pre-intervention, and in red here is the post-intervention time period. And you can see, for example, an expansion in bifidobacteria in individual number 1 that's quite pronounced after the fiber intervention. And we saw a number of really interesting within individual changes where you can use somebody as their own baseline and watch and see how the dietary intervention impacts their microbial community composition. Overall, across the study cohort, we did find that the microbiome composition changes within the 2 weeks of increased fiber intake. And we found that 8% of the microbiome variability within an individual was accounted for by the high fiber diet. And here in this ordination plot, where each point represents a single sample and the dashed lines are connected for the samples that come from the same individual, you can see these interesting changes that are associated with the diet within each individual. When we look to see which taxa were changing in association with the diet, you can see an expansion in bifidobacteria, which is known to be good at especially the first step in breaking down fibers. So we saw an expansion in bifidobacteria that are known to be capable of breaking down plant fibers. Prevotella is another one I mentioned, actually. We did see an increase in Prevotella in a cross-sectional way across the people in the study. There were also some bacteria that are known for typically being good, especially at secondary or future steps in the fiber breakdown, which we don't see. We actually see them going down in some cases after just our 2-week intervention. So that was kind of interesting. We had predicted that the overall diversity would increase by increasing the fiber, especially with this diverse whole diet intervention that had fibers from a diversity of plants. And if looking back through human history, when you look at microbial diversity in hunter-gatherer populations, for example, you see that there's much higher gut microbial diversity. And through human history with agriculture and industrialization, we've seen a decrease in the diversity of the gut microbiome. Of course, exposure to antibiotics and other medicines and sanitation and lots of other forces have led to this decrease in gut microbial diversity through the years. However, introducing a high microbial accessible carbohydrate, fiber diet should be able to promote a diversity of microbes within the gut. So we were very interested to see if that happened in our 2 weeks. But actually, so we had this expectation that the diversity would go up in the post-intervention samples, but actually we really didn't see an increase in diversity within the context of our 2-week intervention. And there's different ways of measuring diversity. This is a diversity index that takes the total number of microbes and also the evenness of their distribution into account. And if anything, maybe there's a little bit of a decrease in those couple of weeks. And so this has led us to think about this a lot and talk to other people who are doing diet interventions and try to learn about how long the interventions have to last in order to see a meaningful change. It's led us to ask a lot of questions about is just changing the diet enough, or would we also need to supply exposure to gut microbes that are able to colonize the human gut? and help break down the fiber. I think these are all really interesting next step questions. We were very interested in Jen Wargo's result that I pointed out earlier in the introduction about the high fiber diet increasing the success of cancer immunotherapy outcomes. So that suggests that the fiber on its own, even without the addition of additional fiber breakdown microbes, could be enough to have a profound health impact. So this led us to, just to think about this as a big regime change, and maybe we're still kind of at the beginning of a revolution. We were also interested to see how these favorite molecules, the fermentation products that are the result of microbial breakdown of the dietary fiber, the short chain fatty acids, we were really interested to see how the levels of short chain fatty acids in the fecal samples were impacted in our study, knowing that they have all these important health impacts. But we actually did not see a big increase in the short chain fatty acid concentrations in the fecal samples from our study. We know that short chain fatty acids are often the result of complex microbial cross-feeding networks. And so maybe in just this 2 weeks, we saw the bifidos moving in, but we didn't see some of the secondary members of these cross-feeding networks have a chance to become enriched yet. But here you can see our short chain fatty acid measurements, which we did with a gas chromatography flame ionization detector instrument in our lab. And butyrate, for example, is a really important and famous short chain fatty acid. And you can see we didn't see, in the fecal samples anyway, a big increase. We also are interested to know if we would have seen the same signal if we had had plasma samples, for example. So there's a lot of interesting next steps that we could take, including a longer intervention to try to see how this would all play out and how changes in fiber in our diet could impact our exposure to these important molecules. We did see some functional changes that occurred in tandem with the compositional changes. So, for example, we found more reads in our shotgun data mapping to the inositol degradation pathway, which is thought to be an important pathway for plant-based inositol breakdown, and has been seen to be negatively correlated with colon cancer. So that was an interesting increase we saw in our study. And just overall, to summarize what we were learning from our fiber intervention study, we found that the high fiber diet significantly shifts the microbiome composition and function in the 2 weeks. Although we think that the short chain fatty acid abundances may take longer to respond, or might require a different type of measurement. Maybe static measurements from fecal samples are not the right way to learn about that. And we've published our results in both a science-focused manuscript and also an education-focused manuscript, which contains the materials that we were using when we were teaching the course. And actually, if you're looking for more fiber recipes, I can really recommend a graduate student, Jeffrey Laternio from Lawrence David's lab at Duke, has a lot of these great recipes available on his website. And I know a lot of people stocked up on beans during the pandemic, which has been a topic of conversation. And so here's a New York Times article talking about just, what you can do with all those beans that you've stocked up on. And so with that, I'd like to transition to another project I'd like to tell you about, which was also led by Dr. Andrew Oliver. And this was when we had the opportunity to study the microbiomes of people who live in Ecuador. And so getting back to this idea that we need to be studying and preserving the microbes that have been with humanity through our history. Efforts like the Global Microbiome Conservancy, where they're preserving the microbes. So we had the chance to collaborate with Dr. John Patton, an anthropologist at Cal State Fullerton, who has been running what is thought to be the longest ongoing field site in evolutionary anthropology in Canambo, Ecuador. And here's some of the people, the collaborators he's been working with, including master's students who come down with him in the summer and who helped to collect these samples. And you can see the people in Canambo, Ecuador. They have a really interesting and diverse diet, and they get a lot of their calories from a beverage called chicha, which results from chewing starchy root vegetables like yucca and allowing it to ferment for a couple of days. And then they get something like 3,000 calories a day. So really a lot of their nutrition is coming from the chicha. And they also have gardens and they do some hunting and gathering. So they have a diversity of nutrition sources. So we were really interested to understand which microbial taxa are present in the human microbiomes associated with the people in Canambo. And we were interested to understand how the microbiome is shaped at the household level, so how cohabitation influences the microbiome, and just how the microbiomes of people in Canambo compare to microbiomes from both industrialized and non-industrialized studies that have been done. So we sequenced the fecal, saliva, and chicha samples from 28 different households. And we've published our preliminary results in a paper in the bioarchive. And actually, Andrew Oliver's thesis is also available online, so you can get more information there. But we had fecal, saliva, and chicha samples from these households. And we found that the samples, the chicha, fecal, and saliva samples had really distinct microbiomes. And on this plot, where each point represents an individual sample, you can see the fecal samples are clustering independently from the chicha and saliva samples. And if you look at these sidebars, we're showing the abundance of different microbes in the corresponding samples. So you can see, for example, there was high Prevotella and also Collinsella. These are both microbes that are more abundant, usually in non-industrialized parts of the world with higher fiber diets. And we definitely saw that here. And we saw, for example, in the chicha samples, we saw a high abundance of lactobacillus, while we saw well-known oral bugs were more abundant in the saliva samples. So, for example, Raffia was quite abundant in the saliva samples. We had a lot of fun just exploring the diversity in these samples. The oral microbiomes have high abundance of Streptococcus salivarius, and that's also true in the industrialized world. And we were able to look at the archaea and the viruses. There were definitely some interesting viruses in these samples, which we were very careful to process in a safe way so that it was safe for the people working with the samples. And we found that when we looked at what fraction of the reads did not map to any databases, from the oral samples, we found, here's in millions of reads, we found that there were many more reads that did not map to any databases compared to the ones that did map to databases. So a lot of the taxonomy is not really being represented here because it hasn't been described. So that's a really important feature, especially when you go out in a part of the world that is less studied, that you're going to be really biased by which databases you're using. That was really true for the gut samples. So the gut microbiomes in this study were dominated by Prevotella copri, and we found that the fraction of reads that were unknown and didn't map to any databases was really high. So here you see we have between 2 and 3 million reads on average that did not map to any databases. So everything we're looking at here should be taken with a grain of salt because it's very much biased by the databases we were using. But we did see that the samples in Canambo were enriched in vanished taxa as they've been described by the Sonnenberg. So these taxa that are more abundant in non-industrialized parts of the world and much less abundant in the industrialized populations. And Prevotella is an important part of that group. And we saw a lot of that in the samples from Canambo. We also found in general that the samples from Kanambo, shown in blue here, were clustering with other samples from non-industrialized parts of the world. And that was driven by Prevotella and Catenibacteria and Treponema, for example. These are all bugs that are enriched in non-industrialized parts of the world, compared with other cohorts from the United States and Italy, or our fiber study, for example. They were also clustering together and apart from the samples in Kanambo. Now, when we were trying to assess the overall diversity, and this was using a marker gene approach, so definitely impacted by databases, we saw that we didn't really see a higher diversity coming from the samples in Kanambo. However, when we were assessing what fraction of the reads were likely to be unknown and not mapping to the databases, we saw that we had a really high fraction of those types of reads in the samples from Kanambo compared to the other cohorts. So that's motivation and hopefully inspiration to include the genomic dark matter in your results. So if we don't look in the unknown fraction of our data, that's where all the discoveries are. That's where the new things are. So it's really important that we, at the least, report the fraction of reads or contigs that are mapping to databases from our studies. And then, consider a database independent analysis approach when you have these, especially when you have these data sets that are super enriched in unknowns. But really every data set is going to have phages, and probably a good third or half or more of your reads, it's possible that they're not mapping to the databases that you're using. We took a database independent approach, and we used a k-mer analysis. So in this case, we used k-mers of length 21. So rather than trying to map to a database, we just assessed the diversity of all the different 21 mers in the samples. And using this approach, we actually found very high diversity coming from the samples in Kanambo, which was interesting. And Dr. Andrew Oliver and I are working on finishing up this study. And of course, you can find some of these results in his thesis, which is available. So to summarize what we learned from our study in Kanambo, we found that the Kanambo gut microbiomes are high in abundance of the vanished taxa that are more common in the non-industrialized parts of the world. We found that many of our reads did not map to existing databases, and that database independent analysis helped reveal that diversity. Now, just a couple little small vignettes I wanted to share at the end here. So we talked about different options for how you could manipulate a microbiome. And actually, we have several projects going in our lab focused on phages, the viruses that kill bacteria, and the potential for using them as alternatives to antibiotics. And we have a team in our group that are isolating new phages from sewage samples. We call ourselves the Orange County Phage Team, and we're frequently finding new phages against pathogens and opportunistic pathogens of interest. And because we had this effort going when the pandemic hit, it meant we had sewage samples from every 2 weeks sitting in the cold room, which meant that we could use them to try to study the COVID virus. And so during the pandemic, we've had this effort where we've been collecting weekly samples from 8 different wastewater treatment plants in Southern California with the help of the Southern California Coastal Water Research Project. And this actually represents something like 16 million people here in Southern California. And it's been really interesting. A postdoc, Dr. Jason Rothman, has been leading this project, and we've written a few papers about these transcriptomes that we've taken from the wastewater samples. I mean, it really shouldn't work. COVID is an RNA virus, and normally we're also careful to preserve our RNA before trying to do any transcriptomes. But in this case, after sitting in toxic wastewater, we're actually pasteurizing the samples for safety, and then we're extracting the RNA. So there's no careful RNA later going on here. This is actually a pretty harsh environment. So we were very happily surprised by the nice data that we've been able to get through these transcriptome efforts, which makes me want to leave you with a question. So what virus do you think is the most abundant in wastewater? And you might initially expect that it would be a gut phage. That's an answer I would support. But actually, it's plant viruses. So pepper mild mottled virus, for example, and tomato viruses are really common. And it turns out that, people are eating a lot of peppers and tomato sauce, and they're not always using the freshest vegetables when they make those sauces. So maybe that's part of why we have such an abundance of those plant viruses. Okay, so a couple of future and take-home messages. That fiber could play a really important role in maintaining gut health, even our responses to cancer immunotherapy and cancer treatment, for example. And that we think you need both the microbes and the fiber so that you have the microbes present that can break down, do the microbial fermentation and produce healthy molecules like the short chain fatty acids. And so what this means is that we should all be aiming to increase the diversity and the total quantity of plant fiber and fermentation products that we're getting in our diets. Okay, so with that, I would like to acknowledge and thank the members of my lab who made all this work possible. And I'd be happy to take questions via email or other methods. Thank you very much.